Hello, welcome to another edition of CNA's Opus. My name is Rich, I'm a certified nursing assistant. What I really want to do is I want to get through this really quick, and then I'll go on to some things on cystic fibrosis that I said I was going to talk about, because they kind of go hand in hand with the lungs, the bronchi that I'm studying right now. So, also what I want to do is I'm not going to dumb it down, but I'm going to take some stuff out of it that I don't feel is necessary, because this is hard enough. I want people to understand, and I'm learning, biochemistry and stuff like that. But that doesn't mean that the rest of the world has to, in order to understand some of these concepts. So I'm not dumbing it down. I'm just simply taking out parts that aren't necessary. So anyway, this word is kind of a necessary evil here, but I'm gonna have to bust it out. It's actually one I got at Staples. So Martha Stewart for. It's pretty cheap. It's like $5. So I guess you get what you pay for. But anyway, that right there is the lumen in the center there, and in green there, that's the lower cilia, and then you have the columnar epithelial cells right there, and in black, those are the goblet cells that are secreting the mucus under the cilia. And outside of that, in blue, if you can see it a little bit, that's the basement membrane, and that there is attached by these integrins, which are adhesion molecules. And then outside of that, you have the lamina propria. And in the lamina propria, you have these mast cells, and those mast cells are part of the immune system right in there. And this up here is little capillaries and arteries. Those little snake thing projections. I can't point to them. There's little snake-like projections in there. Those are little capillaries and arteries. And outside there in red is the smooth muscle tissue. So if we go down here, those little snake-like projections in there are those little arteries. They're tiny little things. In fact, these capillaries right here that branch off are so small that it's only about one cell that can fit through it at one time. So one blood cell can go through those capillaries at any one time, and they branch off from here. This artery ends right here into the capillaries, and then there's the capillary ending point right there, the post-capillary venule. Post-capillary venule. So these capillaries and these post-capillary venule are going to be very important in the immune response right here. So the immune response here has the early stages, and then it has the late stages. And I'm not really going to go into a huge amount of details into it, because it's going to really bog it down. So I'm just going to stick with histamine, because that's one of them that's in the very early stages of the immune response, which happens within minutes of an immune response within the lungs, whether it be from bronchitis or cystic fibrosis or asthma, anything like that. But what I want to show really at the end here is kind of the dangers of it. So here you have this 7-membrane alpha helix, and the 7-membrane alpha helix, that's a G-coupled protein receptor right there, and there's 800 of them, which are grouped into 5 families. And that H1 right there takes up the histamine, and this is into an epithelial cell. This is really important. That's taken up into an epithelial cell, and that is going to make translate and transcribe nitric oxide and prostaglandin. So we have nitric oxide and prostaglandin, which this epithelial cell as an intermediate here takes up the histamine, will then release the prostaglandin and the nitric oxide onto the smooth muscle tissue here, and the smooth muscle tissue will relax. It'll relax, and this will cause a vasodilation right here. on the precapillary portion of the ventricle. So the blood is moving in this direction towards the capillaries, and there'll be a vasodilation right here. And it's because of that intermediate epithelial cells that are lining this ventricle. Right? Now here's where it gets really crazy. The same seven membrane alpha helix, which is again a G protein coupled receptor, 7-membrane alpha helix, that there the histamine will hit it right directly on the smooth muscle tissue. Directly onto the smooth muscle tissue. No intermediate man. No intermediate man. Directly onto the smooth muscle tissue. And it has the exact opposite effect. The exact opposite effect. Because what that is going to do is that'll get taken up and that'll transcribe and translate something called that's inositol 1,4,5 triphosphate. So we have on this carbon 1, 2, 3, 4, 5, 6. So that's a 6-carbon ring, and that's an inositol ring is a 6-carbon ring, which each carbon is going to have a hydroxyl group off of it and a hydrogen. So there's carbon 1, 2, 3, 4. It's going to have its 4 bonds right there. And it'll have its 4 bonds right here. And then this, with the exception of the phosphate groups, the rest of them on inositol rings are going to have hydroxyl groups and hydrogen off of each of the other carbons other than the ones that the phosphate groups are attached to. So that's 1, 4, 5 triphosphate, because there's 3 of them. There's 3 phosphate groups attached to it. And that goes to something, you can see it in there in the red blood cell, what does look like one, right? The smooth muscle tissue right here. And that little black spot right there is the endothelial reticulum. And in the endothelial reticulum, it's in the smooth muscle tissue, it's quarantine or holding calcium. It's holding quarantine calcium. So this inositol 1,4,5 triphosphate is going to release this calcium into the smooth muscle tissue, and that's what's going to, the calcium constricts in certain ways by certain mechanisms that I'm not going to go into. They're going to cause a constriction on the smooth muscle tissue. So now we have the exact opposite by histamine. When histamine is released in the early immune response, you have the early immune response and then you have the late immune response. So the early immune response, one of them, and there's a few other things that are released too, but histamine, I'm sticking with that just to ease your mind. Just to ease your mind. Sticking with histamine. Okay? That's in the early immune response, and that's within minutes. Over here on this one, it goes to the epithelial cell first. That transcribes and translates the prostaglandin and the nitric oxide, which is released from the epithelial cells onto the smooth muscle tissue, and that relaxes it. Relaxes it. So this more blood can flow through this now because there's more room. Dilated this. That's vasodilation right there. It dilated that. So more blood is now moving through this. And during the immune response, these little epithelial cells, which have different ways of kind of changing form and moving a little bit, when I get into videos on invatopodia and stuff like that, you can see how cells can kind of shift and move around, stuff like that, but I'm not going to show it right now. So this will cause a little bit of an opening, right? So there's a little bit of opening in the capillary portion and the postcapillary venule, right here, down here. little bit of openings on those epithelial cells. So this blood's rushing through right now. So there's a leakage here and there's a leakage here, which there's pressure on it. So it's opening up a little bit more. But it's creating enough space for white blood cells in the early immune response to come in, which in the early immune response is neutrophils. And neutrophils are white blood cells that aren't really, they're white blood cells, but they're like, they aren't the most potent form of white blood cells. I mean, they're kind of like the pawns in the game. They go in there in mass numbers and start attacking, but they're kind of limited on their attack, and that's in the early immune stage, the early stages of the immune response. Over here, the exact opposite thing happened on the smooth muscle tissue when histamine hit it. It constricted. It didn't relax because it didn't have that intermediate epithelial cell. So the same molecule had a different effect on different cells. And that's important. That's important. So, the later immune response, which I'm going to go into, which could happen in hours later, is the tumor necrosis factor 1 alpha. Now, tumor necrosis factor 1 alpha, if nobody specifies, there's other forms of it, but if nobody specifies the alpha, then you assume it's the alpha. The tumor necrosis factor 1 alpha starts being produced by the epithelial cells here in the later immune response. Okay? And that tumor necrosis factor 1 alpha kind of has a chemoattractive to helper T2 cells, and they'll be brought up and they'll have cytokines, which again are they produce them, and they too are chemoattractive, which will pull in, gravitate in, and activate one of the most dangerous forms of white blood cells. Because in this white blood cells, and that white blood cell is eosinophil. I'm sorry, I just went blank for a second. That's eosinophil. The eosinophil right here, which I drawn, kind of has the look of it, has two lobes connected. I can't point to it. Two lobes connected by a tube right there in its nuclei, and these down here are granules. There's no phagocytosis in eosinophils. Like phagocytosis is like a macrophage. There's an invagination, and then it engulfs the pathogen or the bacteria or whatever it is. These eosinophils have toxins which are released onto the parasite, say for example. Now, if there's an immune system, if there's an immune response where there's no pathogen present, like with an allergen, something very dangerous can occur in the later stages. And the reason it's a later stage is because the tumor necrosis factor 1 alpha, it takes longer to, it has to, during the immune response, it has to be transcribed and translated right then and there, as where histamine is ready to go. So histamine's, once that immune response happens, the histamine is ready to go. It doesn't have to be manufactured. Tumor necrosis factor 1 alpha during that initial stage has to be transcribed and translate it, and then it's slow acting. So that has to occur first. That's why it's in the later stages. It has to be made. It has to be made during the immune response. But that essentially, after a chain of events of bringing in cytokines and attracting in and activating eosinophil can go in here within the lumen here. And if there's no parasite, there's no pathogen, there's nothing. It's just maybe an allergen, whatever. It's nothing. All that. There's nothing there, right? So these eosinophils can actually just start releasing their toxins right here onto the epithelial cells. So you have maybe a destruction of this epithelial cell, and this epithelial cell. Now, and you can actually start destroying parts of the basement membrane here, right? Then you get cells that are within the lamina propria, which get into the lumen, right? And that can start setting off non-allergic reactions, because you have cells just floating around here in the lumen. So things like I don't know, cold air, a bunch of non-allergen reactants hit these cells here that are in the lumen that aren't supposed to be there, and they set off chemical signals saying that there is an immune response right there, because there's cells there, there are neurons there that got there into the lumen. They're not supposed to be there. So something hits it that triggers off some kind of epigenetic shift, and they set off signals saying that something's wrong. And then you can get a non-allergic response. But the big thing that I want to show here is this eosinophil. It gets in there and it starts destroying parts of the bronchi when there's nothing, there's no pathogen there. And that's dangerous. Particularly you start going all the way around here to different bronchi, and it's left untreated. When it starts getting to the stage where things are left untreated and eosinophils are getting within the lumen of the bronchi, and they start destroying the basement membrane and the epithelial cells here and everything like that, that's when things can get dangerous for long periods of time. Over the long term, you're destroying basically the bronchi from the inside via your own immune system, via probably one of the nastiest white blood cells. And nastiest, I mean probably the one that can protect you the most, but also the one that can destroy you the most, is the eosinophil. And just food for thought, they call it eosinophil because of the red dye. You place, what is it? Eosin? Yeah, I think it's Eosin. Yeah, you place it on there, on the cells, and it'll really stick out. Cytoplasm will really stick out in this red color to differentiate it from the other cells that are there. So once that the later immune response happens and that tumor necrosis factor 1 alpha is produced and a chain of events happens and it draws in and activates the eosinophil, and it gets in here within the lumen of the bronchi, it can just start destroying the bronchi from within when there's no parasite or anything there to destroy other than your own epithelial cells. That being said, I did say that I want to speak a little bit on cystic fibrosis, and cystic fibrosis is an autosomal recessive disorder, which means both parents have to have a copy of the allele. both parents have to have a copy of the allele, so they're heterozygous. They're heterozygous for cystic fibrosis. They just have the mutated allele on chromosome 7, I believe it is, and a CTFR. Yeah, CTFR allele on chromosome 7, and if they're heterozygous for it, and they have a progeny, the progeny, if both parents are heterozygous for that allele, the progeny has a 25% chance of getting cystic fibrosis. And you heard me talk about yesterday about how the cilia is supposed to cleanse out the mucus. It's supposed to cleanse out the mucus. And what happens during cystic fibrosis is that the salt is leaving and the water is leaving via these little aquaporins within the cell. So there's no water there to dilate the mucus, so it becomes like this clump, and it's too much for the cilia to cleanse and move. So it just builds up and builds up and builds up and builds up and builds up. This is also another way that this lumen here within the bronchi will get smaller and smaller and smaller and smaller and smaller, because it's not, the mucus is getting, the goblet cells right here and other peripheral cells out here are producing that mucus that's going to go onto the cilia right here. It's not getting diluted by water, so it just gets clumped, and the cilia, it just can't move it. So the mucus, just draw it in here real quick, starts building up and building up and building up, and it makes the lumen here, the air passage, smaller to breathe out of. Smaller to breathe out of. So that not only happens in the bronchi, but also happens in cystic fibrosis in the digestive tract and the pancreas. That is an autosomal recessive disorder again, and there are genetic testings that can be done, which brings up a lot of ethical issues, because if two people want to have children, they can go get genetic testing done to see what's the chances of my child getting this, that, or this, and that's something that you really want to bring up to your doctor and discuss that, because that could really bring up some issues in relationships if the two want to have children with one another. So that, I believe, is it for today. So is there anything else I want to say? No, I think that was it. Alright, Rich, see you later.